Thanks, Ed. So yes, let's get to it. First with the disclosures. So in addition to support from NIDA, which is actually the sponsor of this conference through our NIDA P50 Center grant, I have had projects, collaborations with industry, but solely related to developing less harmful tobacco products. I want to cover three main topics. First, research findings relevant to the issue of smoking and how it interacts with stress, anxiety, pain, and kind of give a personal tour, personal perspective on that through my own research over the decades. Then discuss potential brain mechanisms and then therapeutic implications. So there are a number of methodological considerations that one has to take into account when you're doing particularly laboratory studies of smoking, nicotine, and pain, stress, anxiety. One thing that one has to decide is what state of deprivation, if any, the participants would be in. On the one hand, if people come in, let's say, overnight deprived or deprived from smoking for a longer period, they will be in a state of withdrawal, which is going to then perhaps add to any negative mood that they experience during the manipulations. On the other hand, if you have them non-deprived, then the effects of your nicotine manipulation are going to be attenuated because you're just pushing the blood or brain levels up or down a little bit because they already have levels. So that's always a tricky decision, varies from study to study. Another methodologic question is when would you administer the nicotine or smoke? In a way, ideally, you might do it during a task, but then you have sort of competition between smoking and doing a task and so forth. So often we would administer nicotine or placebo immediately before a task, but that's another decision. Another kind of big division methodologically between studies is whether they look at the effects of smoking on reaction to a stressor or conversely the effects of a stressor on how rapidly people smoke or how much nicotine smoke they take in. And lastly, there's a wide range of stressors that one can present. Cognitive challenge like in-back tasks or vigilance tasks, pain, social anxiety, or the threat of something bad happening. And in that regard, I will take my little walk down memory lane by recalling a study that my friend and colleague from, again, 40 years ago, Saul Shiffman, who's a very prominent researcher in the field. Many of you have seen his publications on the relation between negative affect and smoking. And way back in the early 80s, Saul wanted to do a study of the effects of the threat of shock on people's smoking behavior. And he submitted an application to the IRB saying that, we would present the threat of shock, but not actually shock people. And the IRB objected, saying, well, you're telling people you're going to shock them, but then you don't actually shock them. That's deception. You can't do that. And Saul's response was, well, my goal is just to look at how anxiety affects smoking. We don't actually want to needlessly cause pain. And then the IRB said, well, can't you just shock a few of them? But in the end, Saul won the argument and was allowed to do a study looking at just the threat of shock. When I talked to him, I said, Saul, people are so afraid of electric shock, all you need is a little shoebox and two wires, and the sight of that will get people pretty nervous. So this was my suggestion. But what Saul actually did was something that looked more like this. And the first subject who came for the study, I was kind of looking from distance, saw the gauge, said, that thing's calibrated in kilovolts. He ripped the things off his wrist and ran out. And so that was the end of that study. Meanwhile, I was pursuing more milder forms of stress, and in this one study, had people expose them to a sort of a social anxiety task where they had to compose a comedy monologue a la Johnny Carson, who some of you younger folks may not know the name, but anyway, he was a famous comedian. And then another task was to count the number of digits being read through a tape recorder. Again, a tape recorder is something with a little reel of tape, but anyway. And then there was a relaxation condition where they could just sit back and read magazines. And we found that in terms of the profile of mood states questionnaire, the tension scale was higher in the so-called stage fright condition and the concentration condition than in the relaxation control, as we hoped. And in terms of smoking, they did puff slightly more. So the contrast between the stage fright and relaxation control, they puffed about almost 0.3 puffs per minute more, which over a cigarette means a couple more puffs from a cigarette. And similarly, the intense concentration elicited more smoking. Now, Saul Shiffman has gone on to do a number of studies, and in one study found there was very little association between the rate of smoking or the probability of smoking during the day and stressful, unpleasant affect situations. But he was studying heavy smokers who were basically smoking all the time almost, or very, very often, so it was hard to detect much of a change. On the other hand, when people quit smoking and they're abstinent and relapse, that he has found has strong relation to negative affect, and in particular, a suddenly rising negative affect will likely precipitate relapse on that day, much more than just steady background negative mood is kind of a sudden crisis. Now, turning it around, looking at the effects of smoking on the response to stress, we did this one study in Murray Jarvik's laboratory looking at 4 different stressors that included cold pressor, ice water pain, and another concentration type task, and then a difficult anagram task. That was another interesting interaction with the IRB, because originally we wanted to do unsolvable anagrams and give people a failure experience and then let them anticipate, a repeat of that. But the IRB said, well, we'd have to tell people we're giving you an impossible anagram, which of course then people would give up and then it wouldn't be stressful. So instead we gave very difficult anagrams of words from a medical dictionary that none of us knew what they meant. So that wound up giving people a failure experience, but technically they were solvable, and I think there was one ringer who got them all. If Ed will remember this too. So what we did find is that smoking in anticipation of these 4 different stressors, which, as I just mentioned, there was the cold pressor, and then there was also a white noise, 100 decibel blasts of white noise condition. We did see that in the anticipatory anxiety before the tough anagrams situational stressor, that smoking did reduce their anxiety compared to the control condition of not smoking. And in this study, everybody smoked a cigarette at the beginning of the session, so they were minimally deprived. In another study, which Ed Levin and Frederic Bem, sitting at the back, who played a major role in all of our studies and building the program over the decades, we subjected people again to the unsolvable anagram stress, but looked at the role of the sensory cues of smoking along with the nicotine. So we had a procedure, we were working on early forerunners of e-cigarettes in those days, and we had an aerosol that was very, very, very low in nicotine or tar, but did deliver some of the sensory characteristics of smoking. And we compared the effects of that to real smoke with nicotine and or to nothing, and found that there was a relief of anxiety in both the sensory but very, very low nicotine aerosol and in the real cigarette smoke condition. So there was a role of these conditioned cues. Similarly, in kind of a parallel way, we looked at physical pain in response to a thermal probe, hot probe, with Jim Lane, who is sitting over here in the second row, another longtime friend and colleague, and found that when we measured the tolerance, threshold and tolerance, but the tolerance to a thermal probe which kept ramping up its temperature up to, I think, 49 degrees centigrade at the maximum, that in the three conditions we had, which were abstinence on the one hand from smoking, smoking a normal cigarette with nicotine, and then smoking a denicotinized cigarette, which again gave a lot of the cues, but without much nicotine. And the sensory condition was more or less intermediate between the nicotine and no nicotine conditions, which were significantly different from each other, but this was just a trend, but somewhat suggestive, again, of the role of the sensory cues. I want to mention one study from another Duke colleague, Joe McClernand, who I haven't seen yet, but maybe he is here, just to show that withdrawal is not necessary for showing affect-improving effects of nicotine, because he recruited people who were non-smokers, but who suffered from negative depressive symptoms, and found that a low-dose nicotine patch compared to placebo patch actually alleviated their depressive symptoms. So from these studies, some tentative conclusions are that, first, pain or anticipation of pain and stressful tasks does generally cause some degree of increase in smoking behavior, and particularly relapse during a quit smoking attempt. Smoking and or nicotine does to some extent alleviate negative affect and increase pain tolerance independent of just the relief of withdrawal symptoms, and that the peripheral sensory actions do seem to play a role along with the central nervous system effects of nicotine. So let's talk about mechanisms in the central and peripheral nervous systems. So when we look at pathways implicated in pain, it looks pretty complicated, and perhaps it's not surprising there would be so many different brain structures and pathways involved, because pain has, not only a sensory component, but also an affective component and a cognitive component. I mean, on the sensory side, you have your receptors telling you the intensity and quality of the stimulus, and then you have your sense of suffering during the pain, and then cognitions like, oh, have I experienced this before? How long is it going to last? Will I be able to endure this if it goes on and on? So all these cognitive, motivational, emotional, and sensory aspects interact, and a lot of the brain regions you keep hearing about in the pain literature include things like the periaqueductal gray, the amygdala, the parabrachial nucleus, the thalamus, insula, anterior cingulate, prefrontal cortex, and so forth, and of course the somatosensory cortex, shown up there. Well, those are very distributed regions, and if we look at where the nicotinic cholinergic receptors are in the brain, they also overlap in a lot of these regions, like particularly the thalamus, which I'll come back to, and midbrain cortex, and so forth. These alpha and betas, if you can see them, refer to the subunits of nicotinic receptors. So nicotinic receptors, you may know or remember, are five-protein subunit ion channels that are activated by ligands like nicotine or acetylcholine. And when they, this is the side view through the membrane and then the top view, and they have different types of alpha or beta subunits that are expressed in different combinations. And when nicotine or acetylcholine bind to the receptor, it opens the ion channel, ions flow, activates the cell, the neuron. In terms of the relationship with nicotinic receptors and pain, she et al. Offered this view that, that in the central nervous system, that they're both the, what are called the homomeric alpha-7, which have all the same subunits, alpha-7, and the heteromeric alpha-4 beta-2 nicotinic acetylcholine receptors act on regions like the dorsal horn, where sensory stimulation enters the spinal cord, the locus ceruleus, a noradrenergic source of noradrenergic neurons in the brain, and the thalamus, and that this activates descending inhibitory pathways that inhibit pain transmission, and as well as other actions to alleviate pain. Yet another view from this study, and you'll get these slides to look at and look up any of these papers later if you wish. Umamana et al. Found that nicotine was able, actually a specific agonist for the alpha-7 nicotinic receptors was able to inhibit pain response compared to, so pain response is going up, and compared to either the vehicle control or when a alpha-7 nicotinic receptor blocker was given, the alpha-7 agonist caused a reduction in pain response. And I know Dr. Damaj is going to talk more about this and alpha-7 in particular, but these authors located the alpha-7 receptor on the periaqueductal gray region that is often involved in pain, and that had a descending effect to inhibit pain response. But I want to come back to the thalamus, because when you look at where the high-affinity nicotinic receptors are in the human brain, the thalamus is a real hot spot. And the thalamus is really right in the heart of the brain, if you'll kind of pardon that mixed metaphor. But it plays a very central role in processing every sensory cue except for olfaction that comes from your body. And it's been known a long time, the thalamus actually plays a key role in modulating pain signals. There are many nuclei in the thalamus that process different types of sensory cues, but what I want to highlight is something called the reticular nucleus of the thalamus, which is a thin shell that surrounds the thalamus that consists of inhibitory GABAergic neurons that really filter what gets through the thalamus and what doesn't. And its area, the reticular nucleus, is rich in alpha-7 nicotinic receptors, it turns out. If the thalamus, which has largely been considered a gateway to the cerebral cortex in terms of what stimuli reach your cerebral cortex, the thalamic reticular nucleus, TRN, is considered the guardian of that gateway. So a pivotal role. And it's thought that attention and selection of stimuli and emotions is mediated, at least in part, through the reticular nucleus. And so it kind of facilitates some inputs and blocks other inputs in a kind of filtering selection mechanism through the reticular nucleus. And indeed, thalamic bursts, which can be triggered with optogenetics, reduce nociceptive behavior. In this study, they injected capsaicin into the paws of the rodents and found that then when touched, there was a withdrawal, a sensitization to touch. But by stimulating the thalamic reticular nucleus, you got a marked suppression of the pain response. The pain response was the low bar because that's the threshold for withdrawal. So the lower the threshold, the more pain. So that was relieving the pain here when the optogenetics was given with the capsaicin. And then it returned back to being sensitive to pain afterwards. So also, there's cholinergic input to these thalamic reticular nucleus neurons, arguing further for a possible role of nicotinic modulation. So the hypothesis goes like this, is in daily life, you're trying to focus on pleasant stimuli, like little kitties and rabbits, But then when you want to try to filter out bad things, disagreeable people, scary grant-related anxieties, and so forth, you need the reticular nucleus to block that. I did have another picture there besides disagreeable people, a rather well-known politician, but I figured that this conference is funded by federal money, so left that out. But nicotine may come in as the shield to augment the filtering capacity of the reticular nucleus, just a hypothesis to consider. And going way back, of course, people have referred to smoking in this Friedman Nature paper going back to 1974 as a stimulus barrier, that smoking allows people to kind of keep at bay unwanted thoughts and feelings. And John Castle and colleagues even linked the anxiolytic effects of nicotine to attentional direction. So he had a stressful task and instructed smokers either to focus on the stressful task or to direct their attention away from the task. And there was only a relief of anxiety when they smoked when they were directing their attention away from the task. When they focused on the task, there was no relief of anxiety. So it could be that there's a key attentional process necessary for getting that anxiolytic effect. Well, let's talk about peripheral mechanisms a bit. So as a smoker inhales, there's a rich set of cues that they feel in their respiratory tract. And some of those cues, the smell, the taste, the feeling of inhaling, may provide sort of a non-specific way to help cope with unpleasant stimulation, like distraction, putting your mind on something that's pleasurable rather than the aversive situation. But there also are, I think, specific nicotinic receptor-mediated effects, and we have some evidence from that from a study with a colleague at Howard University, Ozra DeCordy, and what we did with her, what she did, was to label the noradrenergic and dopaminergic neurons in the brain and look at how nicotine activated them, using as a measure CFOS activation, which is an early gene that gets turned on when neurons, for example, are activated. And the idea was to compare the effects of nicotine, which gets in the brain from peripheral injection, to a form of nicotine pyrrolidine methiodide, which is a methylated form of nicotine that has a permanent positive charge and does not cross the blood-brain barrier. Hence, that would provide peripheral stimulation without the direct central nervous system effects of nicotine. And what she found through the immunohistochemistry, and so the first column shows the CFOS activation in red, and then this middle column shows tyrosine hydroxylase, which is a marker for the noradrenergic neurons. This was in the locus ceruleus of the brain, and then the kind of merged picture. So, and then this is saline, which doesn't cause much activation. Nicotine, and this is just high and low magnification of nicotine, did activate neurons, and some of those were noradrenergic, shown by the fact that there's red and green on some of the same cells here. But what's the key finding was that the peripherally acting nicotine also activated some of the noradrenergic cells in the locus ceruleus. And there's the merged picture of the CFOS activation and where the noradrenergic cells were. So there is some direct evidence that peripheral acting nicotine does activate the locus ceruleus. This was the quantitative result, which showed about as much effect of the peripheral nicotine as the nicotine itself in terms of activating these noradrenergic locus ceruleus neurons. And then, and as we remember from some of the other diagrams, the descending pathway from the locus ceruleus can inhibit pain. Finally, let's turn to possible therapeutic implications. So if pain, and for example, is a driver for smoking, and we will hear more about that certainly from Dr. Dietra and others, then one way to attack that problem is to get better pain-relieving medications or other treatments in general. And in terms of things coming down the pike, obviously there's been a lot of attention to the problems of opioid pain medications and the thousands of fatalities every year from opioids. So there are some advances. Here's one article summarizing the treatment of chronic pain with some non-invasive brain stimulation techniques, RTMS, transcranial magnetic stimulation, and also transcranial direct current stimulation, TDCS, are both being explored as ways to block or reduce pain. And then there's vagal nerve stimulation, which is a peripheral technique, but is also used in pain management. And the vagus innervates a lot of the internal organs, including the lungs, and that's, of course, where nicotine can act directly to stimulate vagal nerve endings. And the anti-nociceptive effects of the vagal nerve stimulation seem to be dependent on its input to the nucleus in the brain, the nucleus tractus solitarius, or NTS, and then its projections onto locus coeruleus and raphe nuclei, main serotonin center, followed by the subsequent activation of descending noradrenergic and serotonergic systems in the spinal cord, including opioid receptors, all of which inhibit nociceptive neurons. And you can read the rest of that if you're interested later. So vagal stimulation may act through attentional mechanisms, act through improving mood as well, and through direct actions on these nociceptive pathways. Then we have actual drug treatments, and there are a number of novel pharmacologic therapies that are under study for relieving chronic pain. I won't go into each and every one of these for obvious reasons of time, but again, there's that review article if you do want to follow up, but there's a whole raft of potential new drug treatments to address chronic pain. And there are novel approaches to anxiolytic and antidepressant drugs that are being studied. There's one review paper on that. And finally, nicotinic ligands are being developed, and those may turn out to have some efficacy. There's been a study for years trying to find nicotine-like agents that have therapeutic effects, and perhaps alpha-7 agonists. And then finally, one could say that if smoking or nicotine does have pain-alleviating effects and mood-elevating effects, well, why not just give people safe forms of nicotine, and or at least much safer forms of nicotine than smoking? And there are a number of products being developed or even on the market, from novel buccal or sublingual tablets that deliver nicotine rapid with more or less degrees of rapidity, and then e-cigarettes, which I could take the whole talk or the whole day to get into all of the issues around vaping, but I won't do that except to say I feel compelled to at least have one slide to try to counter what has been nothing short of an anti-vaping hysteria movement that's swept the country, and that is to quote a JAMA study that looked at the toxicant levels associated with smoking, the harmful and potentially harmful constituents identified by the FDA, and looking at people who are either smokers, dual users, or I highlighted at the right, the pure vapors and then never users. And on most of these toxins, and you can read the fine print later, the vapors are almost indistinguishable from the non-users of any product, any tobacco product. So, I mean, there's really no doubt that there's tremendous reductions in toxins, and this recent outbreak of respiratory illness is almost certainly due to contamination of black market street sources, usually associated with THC or CBD, cannabidiol or tetrahydrocannabinol products, and not nicotine. And there was, if you're interested further, the Duke University Law School had a panel, which Dr. James Davis, sitting in the audience, and I and Dr. Pasek from Duke presented. There's the web link, and presents a lot more evidence about that. But there are many ways of potentially giving nicotine without smoke or without toxins that could be another way to cope with some of the pain, both physical and mental. And I will stop there and take questions. Thank you very much.
Well, thanks, Jed, for a great overview of pain and emotional pain and physical pain and tobacco use and addiction. And we do have a few questions for specific questions about his talk, and then we also have a late morning and the afternoon discussion sections for broader issues. So, any questions or comments?
Given the problems with the opioid crisis, are you aware of any studies that address the possibility of nicotine or nicotinic agonists having an opioid-sparing effect in patients with chronic pain?
That's a great question, and I am not aware specifically of those studies. Perhaps some of our other speakers may be. Does anybody want to comment?
Other questions, comments? Okay. Well, thank you, and we'll have more discussion of this in the next session. Okay. So we have then gone from these introduction to the literature in terms of nicotine and tobacco use and pain systems to now very specific investigations, experimental investigations of that. And from Virginia Commonwealth University, it's my privilege to introduce a leader in the field of nicotine experimental research and its interactions with pain systems, Dr. Imad Damaj.